Now, this topic mainly focuses on the mitosis part of which is the M phase of the cell cycle. The M phase of the cell cycle consists of 4 parts, and I like to remember it in this way: PMAT or PACMAT, whichever way you find easier. So these 4 phases can be memorized with just a formula: P, M, A, T. P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. Each of these 4 phases has key differences that you should be able to identify based on diagrams alone. The first part of mitosis is called prophase. Now, the first thing that you might notice in the dividing cell in the prophase stage is that something is up with the nucleus. It's not what you're used to, and instead you're seeing something else. What I'm trying to talk about specifically are these. These are the chromosomes that have formed. Recall that chromosomes have been formed from the spindle fiber. So in other words, during prophase, the spindle fibers contract and shorten to form these structures called chromosomes. Not only that, you will notice that these cell structures that you have learned from chapter 1, centrioles, they have also started to begin moving to the poles of the cell. What are the poles of the cell? Now, to make referring to parts of the cell easier, the cell is compared to a globe, where the globe has 2 poles, or in other words, 2 ends of the cell. Hence, it is said that the centrioles have begun moving to the poles of the cell, which are the 2 ends of the cell. That will be the key signs of prophase. Another key thing that you may notice about prophase is that the way I drew the nucleus was that instead of drawing it as one solid line, I drew it all as dotted lines. I did this because this shows that the nuclear membrane, or the membrane that surrounds the nucleus, also known as the nuclear envelope, has begun to disintegrate or break down. The second stage of mitosis is called the metaphase. Metaphase, one of the key features that you notice is that all the cells are lined up in the middle of the cell. It's lined up in one straight line, or rather to say, it lined up in a plane. Now, this plane has a very special name since it divides the cell into 2 equal parts, and it's called the equatorial plane. So, the equatorial plane is over here, this line that divides the cell into 2, and you notice that all the chromosomes would line up in this equatorial plane during metaphase. Another key thing that you notice is that these centrioles that we just talked about, they now have finally reached the poles of the cell, and there's something coming out of from these centrioles. Now you notice that these things reach the centromeres of the chromosomes. It connects the centromeres of the chromosomes to the centrioles. That is another key defining feature of metaphase. These little string-like things or fibers are called spindle fibers. Anaphase is the third phase of mitosis, and it happens right after metaphase. One of the key defining features, and this one is very obvious, is that you notice that the chromosomes have now finally split into 2 parts. One here and the other one over here. Now, since the chromosome has split, one of these individual parts is called the sister chromatids. So these 2 sister chromatids originally formed one chromosome. So it is said that the centromere of the chromosome has divided into 2, and the chromosome is split into 2 sister chromatids which are separated. Now, the way that this is split is due to these strings. These strings, turns out, they have become shorter. And since they have become shorter than what they usually were, it causes them to pull onto the sister chromatids. So it is said that the spindle fibers shorten and contract, and this causes the sister chromatids to be attracted to opposite poles of the cells. So one sister chromatid will be going this way, and the other sister chromatid will be going to the other pole of the cell. This is how to identify anaphase. So in a nutshell, do be on the lookout for the chromosome that has split into its sister chromatids, as well as the spindle fibers that have shortened and contracted to pull the sister chromatids to the opposite. Telophase is the fourth and final stage of mitosis, and it occurs right after anaphase. So it occurs right after when all the sister chromatids have finally reached the poles of the cells. The pole of the cell, or in other words, the end of the cell. Now, telophase is also rather easy to identify because you can see that the nuclear membrane is back, or to say, the nuclear membrane has been reformed. Since the nuclear membrane has been reformed, it is said that the nucleus, the two nucleoli, are reformed again. You'd also notice that the spindle fibers are now gone, or rather they have disappeared. Now, here's something special. Because now that these, you can notice here that there's about to be two cells that are going to be formed later on. And each of these cells are later on going to be called daughter cells. So, which is why the chromosomes now also have a special name. One of these chromosomes are now called the daughter chromosomes of each of the cells. Cytokinesis is a process that happens right after mitosis, so don't get confused with this. Cytokinesis is not a part of mitosis, but rather it is totally separate from mitosis. It happens right after mitosis. In other words, it happens right after telophase. Now, from the word cytokinesis, you can see that it contains the word cyto. So from this, you can probably infer that it has something to do with the cytoplasm of the cell. And you are correct. Cytokinesis is the division of the cytoplasm in a dividing cell. And this process is different for the animal cell and the plant cell. And this is mainly due to the presence of a cell wall in the plant cell, which is absent in the animal cell. Now, cytokinesis in an animal cell, you may first notice that the two cells appear to be as if pulling away from each other. Although that isn't the right term, that is how I would describe it personally. The two cells appear to be pulling away from each other. So one cell appears to be pulling this way, and the other cell appears to be pulling that way. And you may notice that this sort of pulling kind of action causes a sort of curved shape to appear here. And again, I remind you, this isn't the right terminology to be using, but this is the way I describe it personally. But when you notice that there's a curved shape here, this curved shape actually has a special name in biology. And this curved shape over here is called the cleavage furrow of the dividing cell. So you may state that during cytokinesis in an animal cell, a cleavage furrow is formed. So, what causes this cleavage furrow to form? Now, recall how I described it as a pulling action. Remember what else also pulls during mitosis? You may remember the spindle fibers pulling the sister chromatids apart, separating the centromeres. Now, it's also a very similar concept here with something similar to it's something very similar to the spindle fibers pulling on a ring structure here. So it is a ring structure going all around the cleavage furrow, and this ring structure is called the contractile ring. And in this contractile ring, there is some sort of fiber-like structure or filament pulling on it. This filament that is contracting, causing the contractile ring to shrink, is called microfilaments. So to sum it up, microfilaments that contract, so let me write it down. Microfilaments, oh, the pen's dead, give me a minute. Okay, microfilaments contracting at the contractile ring causes the plasma membrane over here to sort of constrict, and in the end, this causes the cytoplasm to constrict, and in the end, this forms two daughter cells. In a plant cell, cytokinesis occurs differently. You may notice that during cytokinesis of a plant cell, something has occurred within the cell. You may notice that vesicles have started to form in the center of the cell. These vesicles are crucial in the formation of something called a cell plate. Now, these vesicles will start to combine with one another. So in other words, they start to combine like this, like this, like this, and like this, and so on and so forth. And in the end, it forms one large plate running in the middle or the center of the cell. This plate is called the cell plate. This cell plate will elongate or expand outwards until it reaches the cell wall. And you may notice that at last, a new cell wall has been formed. So, this new cell wall has split the plant cell into two new daughter cells. So one daughter cell is over here, and the other daughter cell is over here. So this cell plate has expanded into a new cell wall which separates the plant cell into two, and it produces two new daughter cells. Now, mitosis is of course a very important process, but what exactly are the importances of mitosis? If you are asked this question, the way you can think about it is that mitosis is the process that technically gives us a continuous supply of new cells. So whenever we are asked the importances of mitosis, you can always think about it in this way. What is the importances of having a continuous supply of new cells for an organism? So let's take a look. The very first importance is that it's important for the growth of an organism. For example, let's say a baby grows into a child. The size of the organism has increased considerably. So there has to be something that's contributing to the growth, and hence that is where the new cells come into play. The second more important part is that mitosis helps to replace dead or damaged cells. For example, red blood cells that are dead need to be replaced. So, where do we get the new red blood cells from? Mitosis is the process that helps supply it. Finally, mitosis also helps to repair damaged tissues. An example of tissues could be our skin. And let's say we were to cut ourselves, mitosis could supply the new cells that we need to repair the damaged tissues, or in other words, the skin. Now, a very common mistake that is commonly made while explaining the importances of mitosis is that mitosis repairs damaged cells. This is not true. Mitosis does not repair damaged cells at all. That's because mitosis does not repair cells. But as I just said, mitosis is a process that gives us a supply of new cells.